So we are now getting ready to venture off into the world of biosynthesis of DNA, which basically means what happens to DNA inside of our cells to basically pass genetic information on from one generation to the next. What goes on? What is the message? What's actually being done on the molecular level in order for this thing to work out the way that it works out? It's a beautiful arrangement that happens that Mother Nature has created to basically allow our bodies to pass this genetic information on from one generation to the next. So this description is going to involve a little bit of everything. It's going to go back and it's going to talk about sugars because look, we've talked about sugars with DNA before. We have these riboses, right? And deoxyriboses. It's going to go and talk about these heterogeneous bases, the ATs and the GCs. It's also going to go back and talk about amino acids and proteins because amino acids and proteins help the DNA do its function, but at the same time, the DNA also relays a signal out to make certain proteins and amino acids as well. So we're going to go through and it's going to kind of wrap everything up into one nice, neat little package for us when we talk about this biosynthesis of DNA. So there's really three parts here that we need to talk about when we concern ourselves with the synthesis version. So number one is going to be a process called replication. So we're going to talk about how DNA replicates itself. Now this is important because when we talk about replication, this is passing it from one generation to the next or from one cell to the next. So we need to talk about how DNA replicates itself or clones itself in a way. So replication is going to be object number one. The next thing that we have to do is we're going to have to talk about this thing called transcription. And transcription is important because when we think about transcription, we think about the word transcribe. And when we think about the word of transcribe, we're probably relaying some type of message to somebody, right? So transcription is going to be very important as well. And then the last one is going to be called translation. So once we make this message, we're then going to have to translate that message to something else. So that way we can give it orders to do something very specific. So these are the three portions of the DNA that we're going to focus on. And again, this is unlike many of the other lectures that you received from us so far because this is really generally theoretical information that happens and we've got proof that it happens, but we're not getting nitty-gritty down into the details of the chemistry reactions that go on. We're just looking at this as a macromolecular perspective. So in a biologist point of view, not in a chemist point of view. So three versions here. It replicates itself to clone it. It transcribes a message to do something. And then it has to translate that message to do something different. So those are the three priorities, and that's the order that we're going to in order to talk about these. And we're first going to focus on our replication portion of these three bullets. So what can we say about replication? Well, like we saw in the previous video, is that when we take a look at DNA, well, genetic information is made up in the human cell. Okay, so here is maybe a human cell. And inside of this cell is genetic information, and that's very important. The genetic information has to be conserved. The genetic information has to be contained, and the genetic information has to stay safe. We do not want that genetic information to go out and become modified or mutate on us and change into something completely different. So we've got this genetic info inside of these cells. And this info is contained in chromosomes. And we've mentioned this word of chromosome before in the previous video and in that YouTube video. And what we maybe didn't mention at that point is that there's 23 pairs of chromosomes. So these chromosomes basically are the carriers. There's 23 pairs of those, and those chromosomes begin to get replicated as the cells begin to divide and new ones come along. Each chromosome is composed of genes. And genes are just fragments of DNA. That's all that they are. So this DNA strand, well, certain fragments of those is called a gene. And multiple genes make up these chromosomes that are then crammed inside of the cell for genetic information. Now, these genes are made up of DNA. The DNA, we said, could be 6 foot long or longer, and the DNA can have 3 billion base pairs in them. So we've talked about how this DNA basically wraps around these histones, which are proteins, and the histones wrapped around each other, and then that creates these chromosomes, and that's how the DNA is packaged into the genes and into the chromosomes that then go into the cells. So we've mentioned that before, and we've talked about that process. Okay, so what can we say at this point? Well, one of the first things that Watson and Crick did was basically take a look at these DNA molecules and talk about the importance of the strand and the complementary strand. And what can basically happen here is that we can take DNA and just kind of cut it down the middle, and we can separate these two strands from each other. And when we separate these two strands from each other, they are templates for new strands. So basically what that means is that when this strand separates, this left piece, this black one, so I'll do it here, this right-handed piece has some base pairs on it. And then we get basically a left-handed piece that has some base pairs on it as well. And these are complements of each other. That means the A's and the T's go together, the G's and the C's go together in a certain order. That's the way it's done. They're basically templates or carbon copies of each other. So what happens is that if we can take this DNA molecule, and if we can rip the DNA molecule apart, we get two pieces, a left-hand version and a right-hand version. And then this DNA molecule can begin the replication process. So we get a new strand that comes in, and this new strand basically has complementary bases that match what is there with the original. And then the same thing happens over here. A new strand comes in, and these have complementary bases that match the original. And what we will find out is that when this replication process takes place, well, this strand is the same exact as this strand. They are the same. So this replication process is conservative. And then this one is the same as this one. So when they begin to make the copies, they are basically making a copy of what was already there. And that's the beauty of the replication portion. So we're going to have to start talking about how the replication process happens. You know, how do we separate the DNA from each other? What goes in and begins this copying mechanism to make these strands with? All of that is the world of replication. We are replicating the DNA molecule, and we're using the original to basically replicate the copies. Now, the good news is that it doesn't have to stop here. We can now take this original strand and the new strand that was made, we can rip those apart again, and the original strand can give us another carbon copy of what was there before, and the copied strand can give us a complementary strand of what was there before. So the original strands can be replicated, and the carbon copy strands can be replicated as well. And this is what creates this endless amount of replication that goes on with a DNA molecule. Now, one of the things that we have to realize is that copies can only be made in the 5' to 3' order. That is the way that we write them down on a sheet of paper, and that's actually the mechanism route of which they get replicated as well. They get replicated from the 5' end to the 3' end. So in one of these routes, maybe this is 5' and this is 3'. Well, this was the original matching strand for that, right? So that means that this is 3' and that is 5'. So what we mean by the copies have to happen in a 5' to 3' order is that out of these two, well, this is the original and we are copying the original. So if we start at the 5', we're actually starting here with 3' on the complementary strand. And that doesn't really make sense. It's kind of harder for the molecule to do that. It can do it, but it's harder for the molecule to replicate that way. Now on this side, this was the complementary of the original. It went from 3' to 5'. So if we start up here at the top, this is a 5' end, and this can go to a 3' end, and we can actually get the replication to happen much smoother that way without any problems at all. So this is a 3' and this is the 5' here. Okay, so 3' to 5'. We'll take a look at that in the next slide. We've got basically a picture in the PowerPoint for you to maybe convey this message a little bit better on why 5' to 3' is so important. So these strands that get created, they're called semi-conservative. So semi-conservative. And they're called semi-conservative because these strands make one copy, and one is the original that was there and one is the copied portion that is there. So it's not truly conservative, meaning all of the originals stay behind, but this is basically a half and half. The original parent chain was there, that got split, and then one strand got replicated, and we called that semi-conservative. The semi-conservative, the strand that I've drawn in red here, this is called the daughter strand. So that is the name that we give to that new replicated DNA itself. So that is called the daughter strand. Okay, so just some more terminology and just some more things to worry with. But in the next video, we're going to take a look at replication maybe a little bit more closely, talk about really the details that go on with the strand itself, and hopefully again you've got the PowerPoint there in front of you, because the PowerPoint will have that nice picture that we're going to show in the next video and kind of walk through the description of what's going on. So stay tuned. We're just now starting the biosynthesis of DNA and how this DNA does the wonderful job of passing from generation to generation.